for this invitation. Thank you, Kostya, and thank you to all the audience. I'm going to talk a little bit about the mechanisms for the building up of a social brain, and we use as a model the domestic chick for that, but also human beings. And the starting point is a phenomenon you are probably familiar with, that is filial imprinting. This is a classical and historical movie showing Conrad Lorenz with his birds, showing the classical following response, and then in the Danube. And if you are not familiar with filial imprinting, it is the learning process by which the young of some species, usually of precocial species, learn to recognize their mother by simply, or social partner in general, by simply being exposed to it for a short period of time soon after hatching. And usually Konrad Lorenz is credited as for the discovery of imprinting, but the phenomenon was very well known in the past. It is fully described in the book of Utopia by Thomas More, and in the academic circle, the discovery of imprinting is probably associated with the name of Douglas Spalding. I suspect Douglas Spalding is not very well known to most of you. Was actually a quite obscure biologist of the Victorian time. There is no even image of Douglas Spalding available, or at least I was not able to find an image. But his history is, I think, quite interesting and a little bit romantic, because Douglas Spalding, coming from a humble family, has the possibility to attend for a short period of time to the teaching of Alexander Bain in Aberdeen. And at the time, the Scottish philosophers were mainly interested in the topic of the origins of knowledge. And he wondered why, rather than exploring the topic of the origin of knowledge, making just philosophical speculation, not to use the methods of natural sciences and try to do experiments. But after a while, he was forced to leave Aberdeen because he was ill. He has tuberculosis. So he moved to the south of France, looking for a better climate. And in Avignon, he met the philosopher John Stuart Mill, who was very impressed by the cleverness and the ingenuity of Spalding, and provided him with a job, that is, being the tutor of an aristocratic family, the Viscount John and Kate Amberley. And he moved to the manor of the Amberley in South Wales, and he started a series of experiments leading to the discovery of what he called imperfect instinct, namely imprinting. Lady Amberley was his first research assistant in Ravenscroft, in the manor of the family. And the romantic part of this history, if you are interested, is that there was a love history between Douglas Spalding and Kate Amberley, with and John Amberley, the husband, agree about that. They were a very liberal couple involved with atheism and the trying to have the right to vote to women and so on. So he asked he posed the problem to his husband. This very brilliant young man has tuberculosis, so no possibility to find a proper spouse, a wife. But on the other hand, it would be not nice. He has not access to the pleasure of love. So, with the permission of his husband, Lady Amberley has this love affair with Douglas Spalding. And the interesting part of the story is that he was the tutor of the son and the daughter of the family, and the youngest one was Bertrand Russell. And Russell, in fact, mentioned in his autobiography Douglas Spalding, and he said he was a Darwinian and was engaged in studying the instinct of chickens, which, to facilitate his studies, were allowed to work havoc in every room in the house, including the drawing room. The final part of the story is very sad because Lady Amberley died because of diphtheritis, which was a very dangerous disease at the time. Soon after, even his husband died, and he established that Douglas Spalding should remain the tutor of the daughter and the son, but the father of John Amberley was very prominent figure in England, 2 times prime minister, started a legal action, and therefore Bertrand Russell was transferred to the house of the grandparents and grew up in this house. And Spalding escaped in the south of the France, and after a while he died. He was just 36 and probably would be recognized nowadays as the true father of ethology if his wife was a little bit more fortunate. You will say why I mention all this story, because, well, you will be not very interested because I guess you don't read Italian, but I wrote, it will be published in a few weeks, a novel about this story and a sort of metaphysical thriller about neuroethology. Let's hope it will be translated in English sooner or later. So if you are interested, you can see my very first narrative attempt. Then after this long divagation, let's start with imprinting. In laboratory conditions, imprinting is studied by presenting the chicks with artificial object. This is a movie from Brian McCabe lab. And you could observe that after exposure to an object, for instance, a rotating red box, the chick develop a strong tendency to follow. He is very happy to run the equivalent of kilometers in order to rejoin the mother. But when he is presented with a novel object, in this case a blue cylinder, you can see the chick didn't move, and therefore the recognition of the novel object and also the fear responses is in a sense the consequence of the imprinting process. For a long time, the group led by Gabriel Horn and Pat Bateson, with the participation also of Stephen Rose at Open University, studied imprinting, and they were able to identify a region in the brain that, according to the more recent nomenclature, is indicated with the acronym of AMM, the intermediate and medial part of the mesopallium, which is visible here, and this is the position of the coronal section. And the idea was that every kind, and that was the idea also of Konrad Lorenz, that every kind of stimulus would be identically effective in producing imprinting. So if there is the mother hen around, the chick would imprint on the mother hen, but even an artificial object like a rotating red box would be identically effective. And the research by the group of Gabriel Orr and other identify a sort of sequence of events following exposure that would involve initially the AMM in the left hemisphere. Then there would be a transfer of information in about 3-4 hours to the right hemisphere. And from there, the memory would migrate to another unknown store that is labeled typically as S prime. I will present later on some ideas, some data, because we think to have identified more recently the position of this S store. But before mentioning that, I would like to mention another problem that emerged at the time. It was the 60s of the previous century, in which Gabriel Horn, after a review of several kind of experiments, noticed that, yes, of course, you can obtain imprinting even with artificial objects like a rotating red box. And if you make lesion to AMM, you have apparently amnesia, that is, the animals are not able to make any discrimination between the original imprinting object and a novel one, for instance, to discriminate between the rotating red box and a rotating blue object. But if chicks are presented with a naturalistic stimuli and we compare the condition of lesion with artificial object and a naturalistic stimulus like a stuffed hen, it was apparent that there was a preference for the stuffed hen even in chicks' lesion to the AMM. That produced at the start a sort of, well, discomfort in the lab and puzzlement because maybe they were wrong and the AMM was not the site of learning. But then Mark Johnson and Gabriel Horn performed some experiments in completely naive chicks, that is, before any exposure, before any imprinting has occurred, comparing artificial objects like a rotating red box and a stuffed jungle fowl, so the ancestor of the domesticated species, and they found that there was a spontaneous preferences, not so strong, about 65%, but significant for the naturalistic stimulus. This suggested that irrespective of the learning mechanism, that is, irrespective of the exposure to the object, there seems to be some sort of innate predisposition for certain class of objects for imprinting. So the old idea of Lawrence that any kind of objects would be identically effective for imprinting is probably wrong. There seems to be predisposition. Certain kind of objects seems to be better in order to obtain imprinting. You can say, well, what is special about the appearance of the stuffed and that make it so attractive to a newly hatched chicks? So Mark Johnson and Gabriel Horn performed some experiment to see what is special about the appearance of the stuffed hen, and they used this disassembled version of the original stimulus, changing just the silhouette, for instance, the shape, and maintaining the general arrangement of the features, or making a single part cluster of features such as the legs and the head and neck region. And what they found was that the canonical version was attractive as much as this disassembled version in the experiment for the spontaneous preferences. So this stimulus, which is not very similar to the canonical one, is attractive enough. But if you disassemble the single features in the cluster, making an object like this one in which the eyes, the beak, and the neck region are completely disassembled and rearranged spatially at random in this way, then a preference for the canonical with respect to the scrambled version for the first part is observed. The interesting part of the story is that this preference for the head and neck region seems to be not species specific. That is, it applied to also avian species completely different, for instance, ducks like this one, and even more surprisingly, to a mammalian species like this one, a ferret, which is in a natural condition a predator of chicks. What is important in order to have the preference is the presence of the head region. The head region defined probably in a very rough and general terms, so general that even the face-like appearance of a mammalian species, even a predator one, would suffice for that. These results have been duplicated in several labs in different ways. These are data, for instance, by Cherepov, Tyunova, and Anokin, Kostya Anokin, who studied both C-force expression and behavior when chicks were exposed to a red box or to a foul, to the naturalistic stimuli. You can see that there is more C-force expression to the box, to the artificial object. I would interpret this with the idea that imprinting on the artificial objects would require, in a sense, more brain work than imprinting on the naturalistic stimulus. And in fact, if you look at the session of training, after training, you look at retraining after 24 hours, you can see that there is a strong preference for the naturalistic stimulus, the stuffed fowl, with respect to the to the box. And even reverse training, that is, this is number of rotation towards the naturalistic stimulus, which is higher in chicks that were exposed to the naturalistic stimulus than in chicks that were exposed to the fowl initially after being trained to the box. Subsequently, and this is the more general reason of interest in this story, Mark Johnson moved to cognitive psychology and in particular to developmental studies in human newborns. And he discovered, actually rediscovered, because the original work was done by an obscure pediatrician that observed apparently preferences a few minutes after birth in human newborns for face-like stimuli. That was measured by simply movement of the eyes in newborns. And Johnson duplicated these results, this data, showing that if you present a very simple stimulus with features inside an oval-like shape, arranged in the canonical way as a face-like or completely spatially reassembled or an empty oval, human newborns show a strong preference for the face-like appearance of the stimulus. So that's mostly the historical part and the background, more or less in the same years in which Gabriel Orr and Mark Johnson performed this research. And if I remember correctly, Kostya was also in Milton Keynes and in Cambridge, Professor Anokin. I was a little more south at Sussex, and interesting for different reason in another problem, associated problem. I was interested in imprinting, but given my background in perceptual psychology, I was interested in motion. And I wonder whether any kind, if you look at the textbooks at the time, they say any kind of stimulus is good for imprinting, but better if the stimulus is in motion. So I wonder, is any kind of motion identically effective to produce imprinting, or, for instance, that's my initial conjecture, the very typical characteristic movement uh, of the broody hand with the movement of the head forward and backward would be special in some way. As you can imagine, the problem is to disentangle the purely kinematic aspects of the stimulus from the shape, from the silhouette. And in order to make this disentangling, we take advantage of a phenomenon which was first studied by a Swedish psychologist, Gunnar Johansson, who attached to the joints of a human figure's point of light and then prepare some movies. And if you look at the single image, if you are not particularly talented in projective geometry, it's difficult to recognize a human figure. But when it is set into motion, it is completely effortless the possibility to recognize the shape and the human figure. And not just the shape, you can recognize also the kind of movement, the kind of action that the stimulus is involved with. So we presented young chicks completely naive, without any experience, just outside the dark incubator, with point light display at the ends, the opposite ends of a runway, and the stimuli were calibrated in such a way that luminance, average velocity, and so on was the same. And these are completely naive. And we presented chicks with stimuli like, for instance, a walking hand or a rigidly moving hand. So the pointer light were simply rotating around the vertical axis, or a completely random motion. This is actually Brownian motion with constraints at the boundaries, because otherwise the point could move everywhere in the universe. Or particularly interesting, this kind of stimulus. This is a scramble N version. Scramble N in the sense that the local vector of motion is maintained. That is, every dot maintains the same movement as in the walking hand, but it is spatially scrambled. So you cannot recognize the shape anymore. You cannot recognize that it is a walking hand. Sorry, it is stopping sometime because of the PowerPoint. Uh, but in a sense, from an anthropomorphic point of view or from the perceptual point of view, this stimulus is in some way perceived as biological. You have the impression that this is a sort of creature of some sort, even if of an unknown nature. Notice that in this case, if you are familiar with psychophysical procedure, you can recognize the shape. This is a very well-known phenomenon, structure from motion, in which you recognize the shape on the basis of purely motion information. So you can recognize, which is a sort of the shape of a hand, but the motion is not the natural biological motion. So what we found briefly is that, yes, as expected, young, naive chicks have a preference for the walking hand with respect to rigid motion and with respect to random motion. But surprisingly, there was no preferences between the walking hand and the scramble hand. So similar to the case of the face-like preferences investigated by Gabriel or John Morton, the preference seems to be not specific of the species. There is no anything which is special of the movement of the hand here. And in fact, we move to another kind of stimulus, this one, point light display of this creature, which is a cat. Obviously, a cat is a predator of young chicks. But when we compare the point light display of the cat, we found that the walking cat was preferred to rigid motion and to random motion, but it was not preferred to the walking hand. So a chick just out from the egg would approach identically happy a walking cat or a walking hen. So that's overall picture we have got. It is we compare the walking head with the rigid motion or with the random motion, and there is a preference for the walking hand. But when comparing the walking hand with the scramble hand or the walking cat, no preferences. They are identically attractive. So my original idea that there would be something special about the movement of the hand was wrong, and in retrospect is clear why I was wrong. Why? Well, consider the fact that chicks have a learning mechanism available that is imprinting, filial imprinting. Is a mechanism by which they can learn the characteristic of the potential social partner, the mother hen or the siblings. There would be no any sense in which natural selection would be able to provide in advance all the information in order to recognize the mother hen or the social partner. What is useful instead is a mechanism to canalize, to guide, to provide what Conrad Lawrence called the innate teacher to the young animal. Because when you are a chick and just out from the egg, there is a lot of moving things in the world. So it is as if natural selection would have said something like that. Look, chicks, ignore things like, for instance, rolling stones, completely rigid objects, and also ignore things like completely randomly moving objects, say the leaves moved by the wind. These are no mom, they are no mother. Pay attention instead to those things which are in between, that is, semi-rigid objects, semi-rigid objects which are visible in this point light display, because if you have a light, say, on your arm in different position, because of the rigidity of the joints, there is a fixed distance between some parts and variable distance between other parts. And this characteristics of semi-rigidity is captured in the movement of all vertebrate species. And it is the kind of information, very general information, that the chick is making use in order to direct attention and then to learn about this class of objects rather than different kind of moving objects. You can say, well, that's an interesting and fascinating ethological story, which has limited interest to the chicks, for instance, or other avian species of precocial kind. But luckily enough, a colleague of mine, Francesca Simeon in Padua, decided to see whether the same story would apply also to the young of our own species, that is, to human newborns. So she presented to human newborns just 40 hours after birth the same kind of stimuli we use with chicks. So no human point light of human figures, but point light of chickens. And it is very unlikely that human newborns have had any experience with chickens at that age. And it duplicated exactly our results. These human newborns show a spontaneous preferences for a walking hand with respect to a rigid movement of a hand or a scramble hand similarly. More recently, we look for something even more surprisingly. Colleague of mine, Luba Papel, who is working in Lyon, has discovered in human infants and more recently in monkeys that if the subjects are presented with images of conspecific that are face to face, as in this case, or back to back, the young infants, but also the monkey, show a spontaneous preferences for the stimuli in which there seems to be social interaction. So face to face with with respect to back to back. Obviously, she was quite convinced that the phenomenon is the result of learning mechanisms. So to check for that, we try to duplicate this experiment in chicks using our point light display in completely naive chicks. So they haven't seen any stimulus before the experiment, and they were simply presented and tested for their preferences for this kind of stimulus, two walking hand face to face or two walking hand back to back. And this is just behavior, so spontaneous preferences in a wrong way for one stimulus with respect to the other. And what we found is very surprisingly, that is, this is time, and this is the amount of preference, this is random choice, and there is a striking difference between male and females. You see, male are completely random. They are interested in the stimulus, but they approach identically the face-to-face or the back-to-back. Females, instead, after the initial freezing response, they show a striking preference. You see here the overall preference index for the face-to-face. So there seems to be an innate mechanism to recognize the kind of interaction which is conveyed very indirectly by this point light display by the face-to-face, and there seems to be also sex differences in this. Okay, come back to the original story. After the research we did with biological motion, we decided to look at the face-like preferences in chicks more deeply. You can say chicks don't have a face. Actually, they have, because if you do some special filtering to an image of a chick head region, you can recognize the typical arrangement with three dots, high contrast, arranged like two eyes and a beak or a mouth in the case of a mammalian species. And we use this kind of very simplified face-like stimulus to look whether completely naive chicks do show any preference for this stimuli. For instance, comparing the face-like to this line-arranged number of stimuli, and we measure both first stimulus approach and overall time spent close to one or other of the two stimuli, and we found that indeed they have a spontaneous preferences. And we perform several control experiments to check for the role of symmetry, and we use this Picasso's like stimuli, and we also look for an alternative idea which was put forwards, namely that chicks and young infants could be sensitive to the number of elements in the upper and lower meridians, because in mammalian species there seems to be a sort of anisotropy with respect to that. But even if you pair the stimuli in such a way that there is a larger number of elements, features here in the upper part with respect to the lower part, chicks do show a preference for the face-like appearance. So they seem to have a sort of structural information for what is a face. We also compared, this was an experiment done with Mark Johnson directly, human newborns and chicks, using this kind of stimuli in which we compare naturalistic face of humans with this white noise, in which, from the psychophysical point of view, the distribution of luminance, colors, and so on is completely matched, and we measure in human infants and in chicks the preference for the two kinds of stimuli, and we obtain exactly the same results. And this led me to investigate the general issue whether there could be available in the young animals of all vertebrate species natural born animacy detector. That is, evolutionarily given mechanisms in order to discriminate and to recognize, in the absence of any learning and mechanism, what is animate and what is not animate, sort of life detector, so to speak. So we embarked in a series of experiments to identify, first from the behavioral point of view, and then to investigate from the neural point of view these life detectors. Show you some example. Well, one possible important life detector was hypothesized originally by Aristotle that say that what is typical, what is characteristic of an animated creature, of an organism, is self-propelledness. Animated creatures are able to move by themselves, whereas inert, purely physical, not animated creatures can move only as the result, for instance, of an interaction with another object, by collision, for instance. So we decided to make a sort of very weird imprinting experiment, very different from the usual, in which chicks were not imprinted on an object, but on an event. And the event was the reproduction of a classical perceptual phenomenon, which is the so-called launching effect discovered by Albert Michot, the Belgian psychologist. So there is an object, object A, which is moving and then stop. And soon after the stop of the object A, object B start to move. And then we measure the preference for the two objects separately. Show you the example of the classical launching effect. So, sorry, there is object A, which is moving, and you have the classical impression of physical causality. That is the impression that object B is moved as a result of the collision with object A. So imagine that you are a young chick or a young human newborn, and you have to decide in this very simplified condition, what is more likely to be mom? What is more likely to be an animated creature? Because one object is moving by itself, is self-propelled. The other is moving as a result of the collision. So is less likely to be an animated creature. The difference is very subtle, and obviously there could be several different aspects of the stimulus that can explain on any preference. So we performed a series of control experiments. Show you some of control experiments. One is simply to invert. Well, the position right-left was obviously counterbalance and the color. The other crucial aspect is what object is moving first and what object is moving after that. So for instance, in this condition, the second object is moving first and the first object is then moving. So both are self-propelled in this case, but there is not any contact, any physical contact. A more interesting control condition is this one. Object A is moving, there is the contact, but then there is a delay. And as a result of the delay, to us humans, there is not any longer any impression of physical causality, but rather the physical causality is substituted by a feeling of psychological causality. It is like if one object is moving and then the other object is disturbed in some way. And so he decided to move independently. That is, both are self-propelled in this case. And there is another more subtle issue to be considered. That is to separate, to disentangle the action, that is the fact that one object is the causal mechanism for the movement of the other from the pure self-propelledness. So in order to distinguish between, we have this control condition in which one stimulus, the first stimulus, is appearing as the result of purely disocclusion in this way. and then there is the collision. So the status of the first object is not determined because you cannot say whether the first object is self-propelling or if something has happened behind the screen making it to move. For instance, it could be a non-animated object which was put into motion by another collision. So what we found was that only in the first original condition of the Michotte experiment, there is a preference for the first object, but not in the other condition, which means that it is not the first object to move that decide for the preference, is not the object initiating the contact, it is not the object that has a causal role. What is important for chicks is, or better, they are attracted in some way by object which has an internal energy source, that is, which are perceived as animated, able to move by themselves, self-propelled. We then do the same kind of experiment with human newborns with a different procedure, obviously using look-in time preferences. So we have very similar kinematic stimuli like this, one in which there is visible self-propelledness, another disocclusion from a screen. So the overall kinematics is exactly the same in two conditions, but you see that human newborns do show a preference for the self-propelled object. Another kind of cue for animacy, very important one, is speed, movement. Probably you are familiar with that because sometimes it happens in the fall, in the autumn, that you are walking around and then suddenly in the periphery of your visual field, something is moving, say something moved by the wind. And then you turn quickly and you said, oh, it looks like as it was alive. What is producing this sensation of animacy? Basically, the change in speed, in velocity. An example is shown here, very simple one, in which red stimulus is moving at a constant speed. And we presented this stimulus to newly hatched chicks, and in comparison with this kind of stimulus, which there is an abrupt and sudden change in speed, in velocity. And we found a striking preferences for the stimulus with speed change with respect to the stimulus with speed constant. You say, well, the speed constant is in some way boring because there is nothing happening. But the point is not the change in velocity in itself. Consider this condition in which the stimulus is moving, and the change of velocity is occurring after disappearance in this way. So we have the change, the novelty, because you see an object which is moving at different velocity, but you have not the possibility to see the very moment of the change in velocity. And in fact, the preference is still there. But if you have another condition in which there is a series of occluders and you presented the change in speed at exactly the time of the occlusion, so you see an object which is a constant speed and another which is changing in speed as in the original condition, but you don't see the exact moment of the change in speed, in this case, any preference would disappear. So it is the perception of the changing speed which is releasing the mechanism of the life detector and allow the brain to say, oh, this is alive. A final example is this one. You know that most of the organisms living on the earth nowadays are bilaterian. And bilaterians mean that they have an organization of the body plan that constrains, in most cases, the direction of movement. That is, there is an axis of the body, and typically organisms move along the major axis of the body. So we did experiment again in young chicks and human newborns by comparing conditions like this, in which an object is moving along a trajectory and maintain the constraint that the major axis should be moving in the direction of movement will be along the major axis of the object. Or another condition like this one, in which there is a violation of this simple constraint. And again, completely naive chicks and similar experiments have been done by colleagues in infants show a striking preference for the conditions in which the movement is along the major axis of elongation of the object. So to summarize, there seems to be animacy detectors in the brain. Very simple mechanism tuned to stationary features like the face-like appearance and tuned to certain kind of movement that are able to tell apart for a young organism just after birth. This is likely to be something which is animated, and this is likely to be something which is not animated. So what is going on in the brain? What are the mechanisms, or at least what is the geography, the topography of the mechanism that would allow this animacy detection in the brain? Ideally, it would be nice if we can provide an answer directly in our species, and we try with human newborns to perform, for instance, EEG experiment of this sort. So we presented newborns 40 hours on average after birth with stimuli like face-like stimulus, non-face-like, that is the upside-down version of a face, or this Picasso's like stimuli, that was the nickname in the lab. And these stimuli were presented using I don't know whether you are familiar, tag frequency paradigm, that is in which the stimulus is presented with a white noise with a fixed frequency, in this case 0.8 Hz. And we look for the EEG spectrum at exactly the target frequency. So something like that. And this is the recording in the human newborns. And this is the face, obviously. And the results are really striking in terms of the difference. This is the tag frequency. And you see that the response to the face-like is higher, much more higher than the upside-down version and the very similar appearance Picasso's like version. You can say, well, okay, these are very young human newborns, but you are not completely sure that this is an innate response, because 40 hours of life is in any case a long time. It could be that there has been some exposure to face, say the face of the obstetrics just at the moment of the birth or other. Yes, of course, this is not a completely controlled experiment of the sort I am going to discuss in a minute with chicks, but we think we have indirect evidence that this is an innately predisposed mechanism, not the result of learning, because of the correlation that we observe between the age of the newborns and the strength of the preference. As you can imagine, 40 hours is the average age of these newborns, because we cannot test exactly at the same moment, because sometimes they are sleeping, sometimes they are crying, and so on. But if you try to do that, that is, if you put in the abscissa the age and in the epsilon the strength of the response, you can see that there is a strong negative correlation, which means that the more potentially these newborns have had the possibility to look at naturalistic face, the less strong is the preference, which makes sense because this kind of preference is in my view, a sort of supernormal stimulus in ecological, ethological terms. You remember the supernormal stimuli of the sort described by Nicotin Bergen. He found that, for instance, a pencil made by red and white stripes is more effective in producing the begging response than a more zoomorphic appearance of the beak of the parent. Similarly, I think that the kind of stimuli depicted here, the face-like stimulus, is different from a naturalistic face, but it has exactly the function of being a supernormal stimulus. It serves to the inexperienced animal to pay attention to stimuli which are likely to be similar to the naturalistic face and to direct the attention to the animal to face-like stimuli in order to learn about these faces by cortical mechanism, by imprinting or by other kind of mechanism that would be available. For those interested in the image part, we look also to the source reconstruction, which is imperfect, obviously, very rough with EEG, but you can notice that the same area at around the fusiform cortex are activated by this face-like stimuli. There is also a lot of activation here in this medial part that we suspect, we cannot prove in human newborns, is actually from the colliculus part, that is from subcortical region. This preference is for face-like stimuli is obviously the basis of the very well-known phenomenon of pareidolia. Show you some example of pareidolia here. People spontaneously, I mean adult, not just the newborn, see faces everywhere in the world. And this is Twin Towers, obviously, with the face of the devil. And my favorite one is this one, which is the very well-known church of the stunned chicken. This is a church in Florida. I think that the architect was not aware of the particular effect that you can obtain when it is seen from a particular point of view. Now, anyway, work with children, with infants, with newborns is fascinating, but has strong limitations in terms of the possibility of control of past experience. And this is the reason why we use as a model the chick, the young domestic chicks. This is the brain of a chick, if you are not familiar with it. And we concentrated our attention to look for the regions that could be involved in this innate predisposition, in these animacy detectors, to the region of the so-called social behavior network. Probably you are familiar with that. These are a series of brain regions which are highly conserved among vertebrates. So they seem to be homologous in the avian, in the mammalian brain, that include, among other, the medial preoptic area, the lateral septum, and the nucleostenia, which are of particular interest here. Show you more or less the position of this area if you are not familiar with the avian brain. And this is the position of the septum. It can be actually subdivided in 6 parts. And this is the so-called arcopalium with the nucleostenia. These are regions that could be considered homologous of the amygdalae nuclei in the mammalian brain. And then we have, obviously, as control condition, regions like the AMM, which is involved in the learning, that is in the imprinting process, or another region which is involved in the imprinting process, which is therefore pallial. I don't use the term cortical for birds because they have no a cortex, but they have in a sense of a cortex, a pallium. And this is the medial striatum, which is very well known because of the studies on passive avoidance learning and other to be involving in learning mechanisms. And the technique we use is probably very familiar to most of you, that is immediate early gene expression using immunohistochemical staining of C-force immunoreactive neurons. So the idea is very simple. To count the number of neurons in different parts of the brains when chicks are exposed for a very short period of time to stimuli which are controlled in such a way that from the physical point of view they are identical, like face-like stimuli, but that they can convey or not convey an imacy impression on the basis, for instance, of orientation or on the basis of the pattern of movement and so on, and to see whether there are areas in the brain which are selectively activated by animacy cues, but not by stimuli that do not convey animacy cues. So we started with something very, very simple, very, very general, and very, very rough, if you want. That is, the experimental chick was presented with a demonstrator, that is an alive chick, or an empty chamber. So there was all possible animacy cues available: stationary, the face-like appearance, the movement, self-propelledness, biological motion, whatever. What is similar in the two conditions was olfactory and auditory information. And in this very general condition, we found selective activation in the experimental group with respect to baseline in the septum and in the arcopalium, but not in the regions which are associated with learning. This medial striatum and AMM. AMM is not shown here. Then we try something more subtle, more precise. That is, we compare an alive chick with a rotating dummy chick. So the appearance is very similar. There is a face-like appearance in both cases, but the kind of motion is very different. It's shown here. You can see the two conditions. So auditory stimulation, olfactory stimulation, the face-like appearance is there, but obviously the movement of the stuffed rotating chick has not the characteristics of biological motion, has not the characteristic of changing speed, has not the characteristic of self-propellence. And what we found in this case was selective activation only in the dorsal septum and in the preoptic area, which was also lateralized, that is limited to the right hemisphere for the septum and the left hemisphere for the preoptic area. No other region seems to be activated by this kind of motion stimuli. And then we try something very, very subtle and very, very specific. That is, we presented chicks with a simple red disc moving at a constant speed or with a speed change. Very, very simple animacy cue. Consider these are completely naive chicks. Completely naive chicks. And for the very first time, they move from the dark and see either a disk moving at constant speed or a disk moving with a sudden abrupt change in speed. And what we found in this case was a selective, very specific activation of the septum, only at the septum in the right hemisphere. In the AMM, there is no differences between experimental and control condition. There is only an asymmetry in the C-force expression, which is very well known. There is more activation in the left hemisphere. This is observed in general. And then we turn to stationary stimuli for animacy, that is, the overall appearance of a canonical hand versus a completely scrambled hand. And again, we look for C-force expression, and this time we have selective activation only in the nucleus tenea, which is analogous to the medial part of the amygdala in the mammalian brain. So to summarize, if we present all the stimuli for animacy, we have activation of the septum and the arcopalium in general. Visual configuration stationary in which there is only the head and the neck region seems to activate specifically the nucleus but not other region. Motion cues only, changes of speed and self-propelness seems to activate in a specific way the septum and the preoptic area. More recently, we look at biological motion, that is point display associated with semi-rigid motion, which is obviously more complicated than simply changes of speed. And in this case, the pattern is very complicated. Because what we found is, first of all, a difference between the sexes. We have got changes in C-force expression only in females, similar to the condition facing or not facing point light display. And we found that there is no activation in the septum, but only in the preoptic area and in the nucleus tenea. So it seems that the biological motion is different with respect to motion with speed changes. But the general lesson for all these experiments is that there seems to be not a unique region in the brain that would act as a life detector. The animacy detector mechanism seems to be associated with different regions in the social behavior networks, which are activated depending on the kind of stimulus, say face-like, not face-like, and the kind of movement, changes in speed, self-propelness, biological motion. And there seems to be different parts in the brain which are dedicated to pick up, to detect these different aspects of animacy. Under suggestion of some colleagues, we wonder whether we can obtain some increase of the detection of animacy by making use of the very famous or infamous, if you want, hormones of love, that is oxytocin. You know that oxytocin have an homologous in the avian brain, which is called mesotoxin. And so we administer in the embryo mesotoxin in the brain of chicks and then intracranially actually after birth, sorry. And we look for their preferences, for instance, for face-like stimuli. You can see here the response to saline and chicks injected with mesotoxin. And indeed, we have got, little surprisingly, I think, an increase in the innate preferences. Now let's turn to the story of the face-like responses. Data with human newborns suggested that there seems to be and presumably innate or little affected by experience selectivity to face-like stimuli. In monkeys, the fact that there are neurons responding to face-like stimuli is very well known as they has been described originally by Charles Gross and Rocha Miranda. By chance, by the way, it was a sort of serendipitous finding, but the attempt to discover face-like stimuli in the avian brain until recently met surprisingly with little success. Andreas Niedertry and Mike Colombo, this in Pigeon. So we decided to look for face-like neurons in newly hatched chicks without any kind of experience. In this case, we are pretty sure they haven't seen any face, but we didn't use, as our colleagues do, naturalistic images of other birds, but we use this kind of supernatural stimuli, that is face-like stimuli of this sort. So we have ovals in which dots arranged as a classical face-like or inverted or line, or just the eyes or one eye, left or right, or only the beak, or this Picasso stimuli to check for the role of upper and lower part in the upper and lower meridian. And the experiment was done in chicks 3-5 days old, maintaining in the dark without any experience with face. And they were presented first with the oval without any feature, and then there was the feature, and we record from single neuron. Can show you some of the recording for different kind of stimuli, but I think it's more convincing if I show you a movie. And you can hear, I hope, the sound of this neuron. And you can see the response to the face is very strong, very intense, very low for the beak, but there is something, again, very intense for the face, no response for the line, very striking response for the face, no response for the uh, Picasso's like, virtually no response. That was a surprise for the eyes. We found some neurons responding to the beak, for instance, isolated, but not for the eye. And most of these neurons respond to the canonical orientation for the face, but have no response for the upside down version. And these neurons are clearly there without any learning because chicks have no any opportunity to be exposed before the experiment to face-like appearance of any sort. They have been in the dark. So this seems to us convincing evidence that our brain is innately predisposed to a sort of face-like recognition. And this seems to be true for all vertebrate animals, I think. There is an interesting part of the story which has to do with the original work of Douglas Spalding and the fact that imprinting and this phenomena are typically associated with a critical or a sensitive period. This is very well known for imprinting. You know, imprinting typically occurs within a window of time soon after hatching, typically a few hours after hatching. You can extend the period, obviously, if the chick or the duckling is maintained in the dark. So we wonder whether there will be sensitive period, not just for imprinting, but also for animacy detectors. That is for this particular sensitivity to the kind of stimuli which are some way optimal for imprinting. If, for instance, you look for preferences for speed changes, which is very strong, very striking in young animals, and compare this preference on day 1 and on day 3, it is clearly apparent that the preference is there on day 1, but on day 3 is completely vanished. So there seems to be a critical period even for this evolutionary given uh, innate predisposition. And the interesting question is that it is possible to reopen them, to have a resurgence of this innate predisposition. My colleague Toshio Matsushima has discovered with other colleagues in Japan that the hormone thyroid T3 can reopen the imprinting process. So if you keep chicks up to day 8 of life in the dark, this usually prevents any possibility of imprinting. But if chicks are injected with the thyroid hormone T3, you can obtain imprinting even at this older age. So we wonder whether the same could be true also for evolutionary innate preferences. So we injected T3 in the embryo and we were able to obtain the resurgence on day 3 of the preference for the speed changes. We can do also the reverse. This, if you use drugs that are inhibitor of the thyroid hormone T3, like the iopanic acid. Iopanic acid administered at 1 day of life abolish completely the preference for animacy with respect to the vehicle condition. So we can reopen not just the imprinting, the learning part, but apparently even the innate part, that is, the animacy detectors that make to direct the attention to those kind of stimuli which are more likely to be a mother or a social partner in general. I mentioned at the start that there was this open question concerning the mechanisms associated with the learning part, that is, with imprinting in chicks. And we take advantage of this story of the innate preferences to try to investigate using fMRI for the first time in a scanner, imprinting in the scanner. And we imprint chicks on a preferred stimulus, like a red object. It was actually a red light, or a non-preferred stimulus like a blue stimulus. And then in the scanner, the chicks were repeatedly presented either with the preferred or with the non-preferred one, those that have been obtained in printing either on the red or on the blue. The idea, very simply, is that for those imprinted on the red, there will be simply maintenance of imprinting. But for those imprinted on the blue, when in the scanner they are presented with the red, which is preferred, there will be secondary imprinting. In this way, you can see both the recall in animals that have stable imprinting, but also the acquisition phase in animals that have started on the preferred and then on the non-preferred, and then they show the imprinting to the preferred in the scanner. And the results are quite interesting, I think, because for the acquisition part, we confirm the classical work by Horn and colleagues, that is, the AMM is the main area involved, together with this in yellow, which are obviously associated with the innate preferences I mentioned, that is, the mechanisms that direct the attention to animacy aspects of the stimulus. Obviously, in green, there are the sensory parts, the visual parts, pulsed, and the auditory parts, which are stimulated in the scanner. And if you look instead at the retrieval, that is, at the imprinting in animals that have been stable imprinting on the red stimulus, other than the activation in the AMM, there is striking activation in the NCL. And so we suspect that the S prime, that is the transfer of information from the initial left AM, then after 3-4 hours in the right AM, and then S prime, we think that S prime is actually NCL, that is the nidopallium caudolaterale, which is considered a sort of equivalent, not actually an homologous, but an equivalent in the avian brain of the prefrontal cortex. Coming back to the innate preferences for animacy, consider that I mentioned innate preferences associated with social behavior. But obviously, you can expect that these animacy preferences would apply identically to other aspects of the environment. That is, there are not only social partners in the world, there are predators, there are prey, and obviously life detectors, animacy detectors would be important also to detect not just the mother, but also to detect the presence of a predator or other. Show you just an example, because we studied newly hatched chicks, again without any experience, with two kinds of stimuli which are associated with anti-predatory responses, show you only the behavioral part. The brain work is still in progress. So a looming stimulus, it is a stimulus in which there is an abrupt change in the retinal stimulation, simulating, say, a predator which is approaching, or a translatory motion of a disc in which there is no change in the size of the retinal image. That could be, say, a predator flying above the chick, but who has not yet noticed the presence of the prey. Show you an example of the response of the chicks in the two cases. The stimulus is actually presented on a screen on the top. You will see on the lateral side for presentation purposes. So this is the looming producing a fleeing response. You see? This is the proper response because it could be a hawk approaching you. And so you have to flee to escape. But then look at the response to a stimulus in which there is just a sweeping stimulus, translatory motion in this way. And when notice, you see that the chick immobilized, he has a freezing response and couching down. Because in this case, the more appropriate response is to be motionless in order to minimize the risk of being preyed upon. There is a second condition, you know about that, in which it could be convenient to have a complete freezing response, which is described for the very first time in this very old book in Latin by Father Athanasius Kircher, who was a Jesuit priest, and he has the very first description of animal hypnosis. That is the tracing of a line on a floor, and this has been described several times. This produces hypnosis in the chicken. Actually, the line and the other aspects are simply theater. They are not necessary. What is producing the tonic immobility response is the fact that you take the chicken with the hand, for instance, and block it. So the equivalent of a condition in which the predator has attacked the animal, and then the only thing that you can do is to pretend to be dead. And this is in fact tonic immobility response. Okay, what you can do with this super chick that could be in any way useful to other parts of neurosciences or useful uh, uh, to colleagues working in and interested in clinical aspects or other translational aspects of behavior and mental processes. Well, we reasoned that this life detector, these animacy detectors that we observe in the chicken forebrain and that we believe are actually present in all kind of young vertebrates, are probably the building blocks for a social brain. Because when you have to build a social brain, the first basic distinction is between, is this alive, it is an animate creature, or it is not alive, it is not an animate creature. So the question is, what happens if for any reason, pathology associated with genetics, an insult on the brain or other, these mechanisms are not there in the young animal? So we try to investigate this in two ways. One is more direct, using our model organism species, and making use of valproic acid. I don't know whether you are familiar with valproic acid. It is a drug which has been used in the past as an anti-epileptic and as a mood stabilizer, until it was found that in pregnant women, is associated with a high risk of having autistic children, autistic progeny, which means that the valproic acid cannot be longer used in pregnant women, but make it of large interest to scientists. So there is a lot of work which has been done, particularly with rodents. What we tried something different. We injected valproic acid in the embryo, in the egg, at about 16 day in embryo. And the idea was to check whether we could have a specific impairment in the animacy detector mechanisms. So we injected valproic acid, and then we look for the preferences of naive chicks coming from egg injected or not injected for scramble versus canonical version of the chicken, or changes of speed or constant speed, artificial stimuli. And what we found is that valproic acid indeed impair in an important way innate preferences for animacy. And the interesting part of the story is apparently valproic acid does not impair the learning process. That is, imprinting is pretty normal. So these chicks can be imprinted, for instance, to a stuffed hen. What they are lacking is a preference for a canonical stuffed hen with respect to a scrambled version of it. They approach both identically, but the learning mechanism is intact. So there is a sort of dissociation between the mechanism associated with the evolutionary given predisposition and the mechanisms of imprinting. This is interesting, I think, but it would be nice if our work would be in any way useful even to our species, and in particular, if not for the treatment, at least for the early diagnosis of autism spectrum disorder. And you know that there is a problem, obviously, because currently the diagnosis of autism spectrum disorder is possible starting from about 2, 3 years of age. If you are a very good clinician, very good neuropsychiatrist, probably you can make a diagnosis at about 2 years of age, but not before. And 2 years is a lot of time, and is a time which is completely outside the kind of mechanisms we have investigated in newborns. But there is a possibility to circumvent this problem, which is studying so-called children of high risk of autism. These are siblings of children that have been already diagnosed with autism. So imagine the condition, which is not common, but there are parents that have had children, and this has been diagnosed as autistic. And they decide, nonetheless, to have another children. And they know that there is high risk that there could be along the spectrum, obviously, which could be very different, again, symptoms of autism spectrum disorder. These families are in touch. There are different organizations in Europe. One is the NIDA in our country, that is organized by our local National Institute of Health. These parents, when they have children, there is a general alert, and they contact our lab, and we have a mobile lab, and we can test them when they are newborns with a mobile lab. And we use basically the same kind of behavioral procedure we develop for our young chicks. We haven't started yet any EEG or FNIRS. We hope to do that in the near future, but for the moment we have only behavioral tests. Behavioral tests which are carried out at around 4 or 5 days of age, which is in our country the age at which the mother is released to to home, and then we can test the newborn. And as I said, we use basically the same test we developed for chicks. Newborns are not able to walk and to move, but we can use fixation times. So we presented our subject with stimuli using classical method in developmental psychology. So, for instance, biological and non-biological motion. The stimuli are actually presented to the left and to the right, and there are techniques to measure looking times to one or other of the two stimuli. Or, other example is shown here, face and non-face, that is an upright or upside down version of the face, again measuring fixation time. We also measure more sophisticated stuff like, you know, probably the story of averted and non-averted gaze, which is very important. Even adult people with autism, they tend to avoid quite systematically direct gaze, eye to the eye. And what we found in comparing individuals, newborns at low risk, say normotypical, with individuals at high risk, is that for face-like stimuli, there seems to be complete lack of any preference for the face stimulus with respect to the low risk that show obviously both in terms of percent of preferences and in terms of number of orientation, the usual preferences. Same also for biological motion. Again, the low risk show a striking preference for biological versus random. No preferences in the high risk individuals. Actually, this is not statistically significant but intriguing. The high risk human newborn, they tend to show slight preferences for the rigid motion with respect to the biological motion. They are not interested in any kind of signs of animacy. You can say, well, that's interesting. It suggests that for some reason, this young creature lack, maybe for biological reason, for genetic reason, these building blocks of animacy detectors. They have no the genetic makeup for that. But there could be another possible explanation, like slightly different. We have preliminary data for follow-up in the same newborns at 4 months of age. And if you compare the condition in the low risk and high risk, it is astonishing because obviously in the low risk, the expectation is that this innate preference would vanishing, fading out, because they are no longer useful. So the newborns, few minutes after birth, have a preference for the face-like to respect to the non-face-like. But at 4 months of age, learning cortical mechanisms subside the innate preferences, and they show preferences for, say, the specific face of the mom and not for the face of a stranger. So the original innate mechanism is no longer needed. But interestingly, in the high risk, you can see that at 4 months of age, they show slight preferences for the face. And they show us slight preferences for the direct gaze with respect to the averted gaze. And similarly, they show also slight preferences, significant actually, for biological versus random, not so for biological versus rigid. So it seems that the mechanism is a delayed mechanism in genetic expression. That is, the animacy detectors are there, but for some reason in people with autistic spectrum disorder, they make their appearance in behavior later on, few months after. But obviously this 4 months is crucial because they prevent the development of the typical dialogue between the mother and the children that you could observe starting from a few minutes to a few hours after birth. And the mother are acutely aware of the fact that there is something strange in these human newborns. So we are trying to develop and to convince a colleague, a clinician, that it would be terribly useful to perform this kind of very, very simple behavioral tests for a screening, a very early diagnosis that there could be something that doesn't work in the usual neurodevelopmental mechanisms in the brain of our species. And I must say, I'm very proud that this has emerged from work from our little humble yellow chaps, the young chicks. This is the very long, too long list of my postdocs that contributed to this story that I told you. And thank you very much to the European Research Council. I left you with a little piece of art. This is an American artist, you know probably, Lucia Ekwernan, that made this beautiful movie of chicks. If you are interested not just on the social part, but also on my work on core knowledge mechanisms which are available at birth, this before any experience, you can have a look at this little book, Born Knowing, in which I tell about my research, not just only on animacy detectors, but also on the mechanisms for number cognition, geometric cognition, and logic to some extent in chicks before experience and learning has started to shape and to mold and to organize their brains. Thank you very much very much for your attention. Thank you.